These tubes here that I'm pointing at are your bronchi coming into the red balloons. There's two of them, actually. There's one back there. That are your lungs. And that what you do is, and actually this is kind of one other thing to point out, this blue plunger-like thing is kind of like your diaphragm. Your diaphragm arches up into your thoracic cavity, and when it contracts, what happens is it flattens out, and the volume of your thorax gets larger, the pressure gets smaller, and air rushes from the outside. Now, what you can't see or appreciate in this is that at the top, the only way the air can get in is if it comes into these bronchi. There's a little hole in the lid, and it connects to these two tubes that are representing the bronchi. To flatten the diaphragm, what you do is push this plunger up here. So when this plunger is pushed, this gets flatter. The volume in the P or V1 gets bigger. The P1 gets smaller, and therefore atmospheric air rushes in through the hole into the balloons, which are like your alveoli, and inflates them. And then when the plunger goes up, the volume gets smaller in the thorax because the arch is returned to the diaphragm, and pressure gets higher in here than in the atmosphere, and air rushes out and goes out the hole. And so I would just recognize this for the lab test as a representation of Boyle's law, and understand what Boyle's law is, and like what the blue plunger represents, the red balloons are representing, and the tubing is representing. Is that any questions on that? Yeah, unfortunately, not being in person, we can't play with this, but most of the time, students like to push that plunger and play with this jar. The other demonstration, which you can do at home if you can find two like panes of glass or small thin panes of glass. I use slides, and that's to demonstrate surface tension. So what I have students do in the lab is take a glass slide, two of them, clean them up, nice window cleaner, get them nice and dry, and then just put a small drop of water on the two glass slides and try to pull the slides apart. And it's really impressive how strong surface tension is that can prevent you from sliding them off or from pulling them apart. Often the only way you can get them apart is to slide them. And you may have experienced this some other time in your life when you're trying to separate something stuck by a thin film of water. And that's what's happening between your visceral and parietal pleura. So to lower surface tension, you can add a surfactant. Well, most soaps, and certainly all detergents, are surfactants. So then if you took two slides and you used, instead of pure water, you used soapy water, you would find it easier to pull those two apart. And so those are some of the demonstrations and this apparatus that we do in person to help reinforce these notions of surface tension, the force of it, and Boyle's law in this case. So I am going to stop sharing this and move on to the lecture questions. And I think in the last lecture, I did, not just an hour ago or so, define it, but just in case, the conducting zone is that part of your respiratory tract, which is going to include your upper respiratory tract, your nose, your nasal cavity, your sinuses, your pharynx, which is the tube that connects your nasal cavity down to your larynx, your oral cavity, and then once you get to the larynx, you're now in the lower respiratory tract, but that's also going to be part of conducting zone. So until you get to those simple squamous cells in your alveoli, it's all pretty much conducting zone until you get to the respiratory zone. So you have to be in the alveoli sacs and alveoli ducts to really be in the respiratory zone. Although, as I mentioned, Ileana's group correctly, there is a little fuzziness right at the end of the terminal bronchioli. We're just going to make it black and white and say bronchioli are in the conducting zone. Okay, so this is defined in the lab manual as well. And then we go to the layers of the mucosal membrane. I went through that last time, but let's, you know what, maybe I have to get another shared screen here. Share this screen and go to the slides. Let's look at the histology. And let's pick one like the trachea, I think. Maybe a little bigger trachea. Oh, well. Yeah, let's try a little bigger trachea if I have one. Something that's a little bigger. Or a bronchiole. I should have looked at this one before, obviously. I don't have any 400 unless we look at the... I don't have any. I know you can blow this up. I'm not sure I can when I share the screen. So it's going to be a little small, but I think you can get the layers in this slide of the, this is the trachea. So here we have let me see if I can even label it.
Professor?
Yeah, are you seeing this slide?
No, we're not. We can see like all your labeled ones, but we don't see the picture you pulled up.
That's interesting. Okay, let's go back and do it again now that I have it. Sometimes it allows me to move around and other times it doesn't when I'm on my desktop, so I'm not sure why. Can you see it now?
Yeah, we can see it.
Okay. All right, so this thin layer is, remember, that pseudostratified ciliated squamous epithelia that lines wherever you have only air flowing. That plus what's under it, and I know this is a bit small because I don't have it magnified quite enough. Right under this would be a lamina propria. I wouldn't expect you to identify the lamina propria in the slide, but you should know it exists. Now, partly I won't expect you to recognize it is because all of this is submucosa. So we're talking about areolar connective tissue versus fibrous connective tissue. And so I wouldn't expect you to distinguish those two connective tissues without being under a lot higher magnification. These big things that are all through here, these are all blood vessels. So over the weekend, I did post a slide which shows the tracheal glands. If you see glands in here, they're going to be in this submucosal layer, which is always fibrous connective tissue, but it can have embedded in it other types of tissues, and glands are made of simple cuboidal, like all glands are made. So in 103A, you probably learned that all glands are made of simple cuboidal epithelia. These are no different. And so how do I know this is a blood vessel? Well, the lining here is simple squamous. You can almost see it's pretty flat, not cuboidal. And there's actually blood in this, and that's what's filling the interior. So unfortunately, the higher power picture, which shows the tracheal glands, is I think I posted over the weekend, so you should be able to find those in there. Now, most of the trachea has its muscularis mucosal layer, or muscularis externae layer, replaced, so it's no longer muscle, it's now cartilage. The exception to that is the trachealis gland that I think I showed you last time. So that muscle, that smooth muscle, bridges the ends of the arch. So if you have an arch of cartilage like that, where my fingers aren't touching, that's where the muscularis mucosa, the trachealis muscle is found. And all of this is muscularis externi layer. And then here's a bunch of adipose tissue, which is in your adventitia. So this is another fibrous connective tissue, but often where you have some structure like this that you need to protect, you cushion it by putting a lot of bubbles or adipose tissue in that fibrous connective tissue layer of the adventitia. So those are the layers until you get into the thoracic cavity itself. And then instead of adventitia, instead of fibrous connective tissue, if this were a bronchi, there would be a serosa on the outside, unless that bronchi is bound to something. And then you might see more fibrous connective tissue. But within the bronchioli and the lungs and such, you're not going to see adventitia. You're going to have serosa there. So those are the layers and the subtypes of tissue. The particular cartilage you find here is hyaline. You unfortunately cannot tell the difference in the larynx. The only other cartilage subtype we'll run into is elastic cartilage, and it looks very similar to hyaline unless you stain it specifically to show its elastic fibers. So you just have to know that the epiglottis is elastic cartilage, not hyaline cartilage. So you can't tell by looking. There's no telling by looking. Okay, I'm going to stop sharing the screen. Are there any questions on that one? Let's see, there's something in chat. Oh, it's not sharing. Okay. All right. So, so let's take a look at some of the pictures of the models for the next one, which talks about the adaptation. And let me see if I've got those pictures open on my desktop. I'll get rid of this one. And sorry. Did I not put the half head picture on here? Respiratory histology. Respiratory models. No, that's not the one. Hold on. I had the Boyle's Law open, so I didn't look for this one. Oh, God, maybe I didn't put that one on too. Let me look in the upper. Oh, here it is. Sorry, I missed the. Okay, I'm also going to rotate this, if it will do it for me. Okay, now I'll share this screen. You're going to get to know this model really well because we're going to use it not just for the respiratory tract, but the digestive tract. And this is a mid-sagittal section through your head. And it does include some, but not all of the sinuses. Now, why you don't have the maxillary sinus, you might remember, is the one right in the maxilla bone above your teeth. And that's because there's two of them, one on each side, and so you can't see it if you're mid-sagittal. However, we do get to see it in that half-head model. So some of the things we're not worrying at this point about the brain here, but right below it, you have the sphenoid, and you shouldn't actually see the ethmoid sinus, which also straddles the midline, but this model went ahead and put the ethmoid and sphenoid sinuses. And then this is the frontal sinus. So you might remember these from the skull, which we could look at if you'd like, but to review, maybe I won't show it to you now, but if you want to, we can come back to it. These sinuses have connections through the bone, through little sinuses in the bone or little passageways foramen in the bone to reach into this nasal cavity. So when you breathe air in, and this area is called the opening itself, the physical gap is called the nares. And there's lots of little vibrissae. You might know men have a few more than women, but women have them too. There's little hairs inside this. This is lined with stratified squamous keratinized epithelia, just like the rest of your skin, and hairs are growing out. They're hair, but we call them vibrissae there. So as the air comes in, these little hairs act as a filter, but also increase turbulence. Remember, that's a big theme here. So turbulence already starts before it even enters the nasal cavity. So these are, remember, some adaptations that you're looking for to help in the warm moistening and filtering air. While making the air go turbulent is going to aid in all of that. The air enters into the nasal cavity, which looks pretty big here. It's more of a canal through the bones, but then opens into this convoluted chamber. And I showed you the horizontal section before. This is a mid-sagittal section. The horizontal section showed you the labyrinth that air has to go through. So even though this looks kind of wide open, it's like going into a maze. And that maze is made as, I think it was Dina or Ileana, I can't remember who said it earlier, because of these three concha that are extensions of the bone into the nasal cavity like this, making the passageways very narrow. All of this is lined by a mucosal membrane with pseudostratified ciliated, and the cilia are pushing the mucus down towards your esophagus so that you'll swallow it. Now, the one exception to that is up here on what's called the superior concha, which is lined with your nasal olfactory epithelia. So all the little olfactory neurons are coating this as well as the pseudostratified. So this concha is a little bit unique in that way. These concha aren't. They're covered with the pseudostratified epithelia, and this one's called the middle one, which is nice and convenient, and this one's the inferior one. So when air enters, it's not only going to bounce around this maze, but some of it's going to go up into your sinuses, which, if you recall from 103A, serve to lighten your skull. That's probably a lot of the reason why you have sinuses. But since you've got these holes in your head, you might as well, to lighten your skull, send air up into them, making more turbulence and more chance for the pseudostratified cilia epithelia, the mucus, to trap any of the dirt or pathogens, and again, moving them through cilia down towards your esophagus to swallow it. So sinuses serve that purpose. They also help you make your sound resonance for speech and aid even in hearing. The skull can vibrate and help you hear with that. So we gave those functions in 103A for these sinuses. Okay, so the air is bouncing all around through all of this stuff. It's getting hopefully warm, moistened, and the mucus is trapping things to filter it. And when this narrows down, so right in here at the very narrow area, it's the beginning of the pharynx, which is going to go down into the top of the larynx, all the way till it reaches the larynx. And right at that location, you're going to see the first of the crypt-rich tonsils. So there's one tonsil here, there's one tonsil here, and I know it's hard to tell, but there's a tonsil right here too. I probably should label the tonsils on these so that, since the slide, this is not very, very clear in the picture of the model, that this is a tonsil. It has a texture to it, if you were there in person. By the way, this is your tongue, and of course your oral cavity, you can breathe through your mouth as well, is stratified squamous because, and so is all of this, because you are sending food, fluid, and air in the common oropharynx. So this is nasopharynx to get to the back here, and then this is oropharynx. Now this little fatty thing that's part of your soft palate, your hard palate has the maxillary bone and the palatine bone here, and then the soft palate, which is this is meant to be like fat, and I guess it's muscle, is called the uvula. And we won't be talking about it much right now. Well, I guess we can. When you go to swallow, when we do the digestive tract, we'll talk about this. This little flap will hopefully reflexively be pushed upward and cover your nasopharynx so that food and fluid go down into your esophagus and not up into your nose. But you know that can have a problem every now and then if you laugh or something while you're trying to swallow, and you know, water can shoot out your nose or something. So good. So we've got oropharynx, which starts right at the uvula here, nasopharynx that goes above it till you get to the nasopharyngeal tonsil here, and then down you go towards the larynx. This is all considered pharynx. So once you get past the oral cavity, it's the laryngopharynx. Okay, any questions on this model? And remember, these are the three tonsils that the lingual, the nasopharyngeal, and the, just blanked out on the third one. Do I have them right now? I didn't name them in my notes, and I've forgotten. I think this one's oro, but I can't remember. So sorry, too much information going by me too. Any questions? Okay, so this is an important model. We will come back to it when we do the oral cavity and digestion. So you're going to see a lot of that model. Okay, let me see. When I stop sharing that, it stopped sharing everything. So, so we talked about nasal cavity, sinuses, oral cavity, pharynx, but we didn't really go into the larynx. So I think we quickly went through the slide of that. And over the weekend after, Friday, I posted a labeled picture, which is a composite between those two separate anterior and posterior histology pictures of the larynx. And so you now have the reference where they're both there. They're actually labeled pictures, so you can see what things are, since there isn't any good drawing in either the lab manual or the textbook, or even online. You'll have trouble finding the larynx. But let's take a look now. I need to go back into the models, and once I do this, I guess I'll close that. And let's take a look at the larynx models. I have quite a few pictures of them. And let's rotate that one again. So this is the larynx. I'm going to share this one. Share the screen. This is the larynx model. We have the larger one that has no soft tissue or very little soft tissue on it. This one has the soft tissue on it. So what I mean by the soft tissue is I think the one that's the gray model, which I won't show, but you can look at, only has the thyroid here. This is the thyroid gland on it, and it doesn't show you some of the soft tissue when I open this up, like the vestibular folds. They're not there because it's basically the skeletalized and whatever connective tissue holds that skeleton together. So it has the hyoid bone. This is that bone that hung down underneath the jaw of the skeleton in the lab. I think you all got through studying the skeleton in the lab last spring before we went into shelter in place, and it was hung by little wires. So it looks substantial here, but it's really a delicate little bone. It's an important lever for swallowing, and so that's its leverage system, but it also acts as sort of an anchor for the cartilages of the larynx. So I don't have you, I know this is hyoid bone, and I don't worry about this ligament that's holding the cartilage here, but you do need to know just a few cartilages. This is the thyroid cartilage. It's the big shield. You can actually feel it at the top of right under your jaw. You can feel that big shield shape of this thyroid cartilage. All of these are made of hyaline, by the way. This is obviously bone, but this is all hyaline. Now, underneath it is the cricoid cartilage. Both of these kind of wrap around, but the cricoid gets a lot bigger on the posterior side, as you're going to see. Now, this little ligament, I don't have you name, but you can actually feel a little indentation where it is. And I pointed out, I think in the lecture, that's where you could do a field, out in the field, tracheotomy. You don't want to go down, not tracheotomy, but where you put the pen in so someone can breathe if their respiratory tract is blocked. I guess that is a tracheotomy. So you're making an opening for air to enter in if it can't get through this passageway, because say they aspirated something into their glottis and it was blocked like food, and you're trying to do the Heimlich and it won't work. Then you may want to cut into the throat to get air in there. Now, in the hospital, they put it way down here, but of course, there are those big blood vessels right there. You don't want an amateur doing that. So, so these rings are then the trachea. So once we get below the cricoid, you're in the trachea. This is the most inferior cartilage. So again, you don't need to know these muscles, or I don't ask about the thyroid gland, but of course that's how this one got its name. So let me stop sharing and open this model up so you can see. Well, let's first look at the posterior view of it while it's still intact. And we'll share the screen. Is this useful, by the way? You can tell me, no, I can do all this on my own. Anyone feel like it's useful? If not, do you just put in chat? I don't even care. Okay, some people say it's useful, but most of you are saying nothing. Okay, so this, I'm giving you guys a hard time. This is the posterior part of that thyroid cartilage. And there's a little bit of the hyoid bone there. And this is cricoid cartilage. So the cricoid, you see, gets a lot bigger on the posterior side. And it not only is a physical tube, it's trying to make an opening that's rigid, this skeletal cartilage, but skeletal, so that this doesn't collapse when, say, you bend your head. If you just had these rings, you might collapse your trachea. So you have this larynx in part in order to, right where your head bends, have some skeletal structure to keep your airway open. And because you've got that, well, evolution has exploited that in embedding into it the epiglottis so that you can direct food into your esophagus. Now, the esophagus would sit right here. It's not in this model. It would sit right behind this cricoid cartilage. So when you're drawing that model, you are going to see cartilage on both sides. You're not necessarily, we don't show the pictures going down enough to see the cricoid. Instead, you're seeing this arytenoid cartilage here. And part of that cartilage acts as an anchor for all these muscles, and those muscles help you move your vocal cords, your true vocal cords. So this guy here is the epiglottis. And if you were to swallow, this would bend down and cover the glottis here. The glottis being actually where air is, the opening. We're going to open this model up so you can see the interior of this model next. Let's see, it's useful. Lots of people are now saying it's useful. All those quiet ones. Okay. So interior view of the larynx. Okay, let's share this one. Oops, I got to move you guys over. I'll move that over. Okay, so now again, posterior back here, anterior here. So this is thyroid, this is cricoid again. And this is actually two, but we're just going to lump them together as all arytenoid cartilage to make it a little simpler. This blue is your epiglottis. So this is elastic cartilage, but all the others so far you're learning are hyaline. And that's the hyoid bone right there. So this is a bunch of fat. You'll even see that in the slide. Because remember I said, and just like in the slide, this is kind of cut away. Your oral cavity would be extending up here. And so you do see this little narrow channel on the anterior side of the epiglottis, but that's all part of your oral cavity, and it's just been all dissected away, as it were, in this model. Here's the epiglottis. It's covered with stratified squamous epithelia because, of course, it needs protection against the food and the fluid that's going, well, especially the food that's going by. And it bends down to meet this area and cover your glottal airway. So when you're not swallowing food or fluid, it's upright, and the air can pass through the glottis here. And as it does, you encounter this is a bump. It actually physically sticks out, but it's soft tissue. So it's not shown in that gray model. It's just not there. The gray model just shows the cartilages that are underneath all those soft tissues. And this is the vestibular fold. I highly recommend that you call it that, not false vocal cords. Call it a vestibular fold because, well, that's kind of what it is. And also, you won't confuse false and true vocal cords because they have different tissue types and they're different functions. So the vestibular folds are like fatty pads that you can, if you choose to, push together and therefore block air passage through the glottis. And that's so that you can increase interthoracic and therefore inter-abdominal pressure. So when you push to give birth, you're using those vestibular folds. It's also true that they make a narrowing of the glottis because even when they're relaxed and open, there's a narrowing, and you can change the airspeed using them as well by narrowing or opening a bit, but that's not their main function. Their main function is just to block the glottis altogether. So it's not that they don't contribute at all to tone of your voice, but if you want to control that tone, you're not doing it with them. You're using your true vocal cords to control the frequency of your voice and the sounds being made. Now these guys, and I know in this model it's a little hard to tell, in this model they just painted them white because that is actually, if you look down somebody's throat, what they look like. Now why they look like that is there's no epithelial layer on them. They're just ligament. So they're dense regular connective tissue that's going across, and you're just staring at the collagen. Very thin, very delicate. These are the true vocal cords that are, what is that, number 28? That are 28. Now in the gray model, they are a piece of felt or cloth, and you can see the model is designed if you could play with it so that you could see how moving the arytenoid would change their length. So there are ligaments connecting these to this cartilage, and there's a bunch of muscles that are no name need to be known that that allow the pulling on this cartilage to change the tension on this ligament, the true cords. Okay, so then finally we get below the vocal cords. By this point, we've transitioned, well, we transitioned somewhere around here, deep in the, underneath the epiglottis, into pseudostratified. So if you were in the lab, and I didn't want you to make a giant composite picture, that's why I didn't do it, you could just keep following this down, and it's going to be stratified squamous until you get down near the base of the epiglottis, and it really rapidly becomes pseudostratified ciliated. So by and large, except for the larynx, the tissue subtype lining this is all pseudostratified ciliated, because only air should go that way. It's just the epiglottis and kind of the back wall over here above the arytenoid that's still stratified squamous. And that's because once you swallow, food should go down your esophagus, which would sit here. Now the esophagus is collapsed when you're not swallowing anything. As you can see in the slide, they're actually touching each other, because its normal position when you're not swallowing is to collapse it down. The last thing not probably good with this model. Okay, let's see if there's another model we should look at before we leave this. I think we did that one. Oh yeah, this is, let's just look down this gray model. The reason I kept it is because, kept showing it to you, is because you do get more of a sense that this piece of cloth are your true vocal cords. And the arytenoid cartilage, you could see where they actually have little strings that are there's a string attaching that cloth, which are like ligaments, to this piece of cartilage. There's no soft tissue here, but muscles would pull on this cartilage, and you could see there's different angles, and that means there's different tension on the true cords. And then they have the little epiglottis on a flexible pin to show you how, if you bend this down, it would cover this passageway. That's the hyoid bone, and we're looking down like we would in a laryngoscope down somebody's throat. Okay, those, I think, let me go back and look at, I think those are all the, I'll just review this one briefly for you guys, if I can turn it up right, because I know 103A was a long time ago for you guys. But these are the sinuses in that half skull. And one of the things that, are you seeing this? Seeing this? Yes or no?
Yes, we are.
Oh, great, because it's kind of small. I don't know why that picture didn't fill the screen like the others did. But so this is ancient. It's probably 50 some years old now, and it's been quite broken. And so I'm kind of disappointed that we didn't get as many of the things that didn't get ordered when the order got canceled on us before we moved to the new building. And I know this shadowing is a little hard to tell, but this is the sphenoid sinus that I'm indicating right here. This one's easy, the frontal, you can see that easily. This is the maxillary. So they went a little off midline so they could cut into the maxillary. This is actually broken, because this is old, and it's part of the inferior concha. So this actually projects out. And then the superior concha projects out here too, but it's a little hard to tell. And if you're seeing little bits, because it's broken again, of the like web-like ethmoid sinus. So the ethmoid sinus is under the superior concha. So this is all part of the superior concha, but some of it's broken away, and you can see the little two circles of some of the ethmoid sinus. So maxillary, frontal, ethmoid, and this is the sphenoid sinuses. So unfortunately, concha, and I could look and see if I have any pictures of the skulls straight on, but if you had those intact skulls, you can see them inside the nasal cavity. Let me stop sharing that and remind you well, this got to go away. Get rid of that picture. From last time, I showed you, I thought it was relief model. I know I showed it to you. So it was that relief model again, but showed you the effects of those concha. What did I call it? No, I'm not seeing it right now. Well, maybe I'll find it while you guys are. The last thing I thought we could do is the lungs are pretty straightforward. Bronchi and trachea, maybe not the last thing. Let me see if this is the picture. I think it is, which I'm pretty sure it is. Yeah. Okay, so let me share this one. So this is a different version. I have lots of versions where there's the larynx. So here's the thyroid gland and the thyroid cartilage and the cricoid cartilage, which is mostly has this muscle on it here. But this main purpose of showing this, are you hopefully seeing this? This model is that it shows you the rings of cartilage become, I'm sure not as quickly as this model is showing you, but they start to become less like a ring. And then unfortunately with the coloring, it's hard to see. Maybe what color one can you see it best? Probably over here. There are little plates of cartilage as you get down to the smaller bronchi and bronchiolae. So the reason why they have plates is, of course, that means that smooth muscle can cause constriction, but that largely happens in the smaller bronchioli, not the big bronchi. Now, I know these are numbered. This is the primary, and then there's secondary, tertiary. I won't ask you about that. The only thing I would know is that we only have two primary bronchi. Other animals, which we're not doing any dissections, but like pigs have three primary bronchi. There's nothing written in stone to say, you only have two major branches going to the lungs on each side of your body. Now, there is an asymmetry to these. You can see the one on the right side of this person's body would be straighter, and this one more bent because the heart sits in this area. And that also changes the lungs so that on the left side, because you have the heart there, you only have two lobes to the lungs, and on the right side, you have three lobes to your lungs. So some of that space in your chest wall is taken up by the heart and the pericardial sac. So I don't ask about, numbering these guys. And I also don't ask the names of the lobes and the fissures and things that are all labeled. You just have to be able to tell me which is the left and the right side of the lungs. And when you go to draw the lung. And you have to tell me something about where these guys enter. They actually enter into the lung around these primary bronchi bases. And that entry area is kind of reinforced with a lot of connective tissue because not only do the bronchi go in there, but the large vessels, the pulmonary circuit enter into that same area. So that reinforcement is, and let me see if I can write it for you, is called a hilum. And we're going to run into this in other body systems too. When we get to the kidney, we'll have a hilum. So this is a area and an organ that is reinforced by, I'll just say fibrous connective tissue in order to support entry of vessels or other tubes. So here the other tubes would be the bronchi, and the kidney, it'll be the ureter. So it's just a generalized structure where you're reinforcing that area. So in the lung models, it's the old models, at least it's labeled yellow. In the redder models, it just looks like an indentation where the vessels and the bronchi go in. The last thing about this model are these colors, and we have, unfortunately, because we're behind in the lecture, what these are meaning to indicate is that once air gets into this bronchi, that's in the, say, salmon-colored bronchi here, the air that this goes to, the alveoli set that this goes to, does not mix air with the ones coming from this oranger color or from the yellow nearby. So these all end in dead-end sacs, like cul-de-sacs, multiple cul-de-sacs, not one alveolar sacs, but a whole bunch of them, a cluster of them. And the reason you want that is so that if you do manage to have a pathogen get as far as your salmon color here, you are going to potentially infect this lung tissue, this set, but it's going to be a lot harder for that pathogen to go upstream and go in the wrong direction to get into the air sacs over here. So ideally, it limits the spread of these pathogens. All of the lining of these is all pseudostratified ciliated, but the cilia now are pushing the mucus up towards the esophagus to swallow it again. If you get something past that pseudostratified ciliated, it is in you for the rest of your life. So if it ends up in the dead ends of those alveoli, then you got it. It's not leaving. So we'll may talk later about it. One last thing, this crux right here is covered with a lot of sensory neurons. Excuse me a second. I've got a whining dog. I neglected to mention all along your upper and lower respiratory, besides the pseudostratified, you are going to have lots of tactile sensory cells that pick up irritations. And this area, which is highly enriched with those, is called the carina. Let me spell that. It's like the woman's name. Area at the fork, maybe, of primary bronchi, trachea, with lots of extra tactile neurons, tactile sensation. And if anything were to trigger it all along this, but especially there, it's very sensitive. It's going to cause you to cough. And in fact, if it gets to the carina, you'll be coughing violently to try and expel whatever comes down there. And of course, along the upper tract, you could cause a cough too, or you could be sneezing. Again, all of those things are meant to be helping you filter so you can expel these things. What are we doing on time? Okay. All right. So let me stop sharing this. And the other one I wanted to spend a few minutes with were the, oops, wrong one. Oh, damn, I keep opening that. Here are the models, was some of the muscles. I just wanted to review for you. Let's start with this one. I got to turn it around again. Okay. So this is all from 103A. So, I mean, it's a nightmare that's returning to you because you have to know some of these muscles. I am not going to include all respiratory muscles. So like these muscles, we didn't do 103A, so I'm not going to include them in B either. But this muscle, remember, connects the ribs together. When you exhale, you want to decrease the volume. So if you bring your ribs together and during exhalation, and I'm not talking about relaxed exhalation because remember that just means you're relaxing muscles that had contracted. This is when you're blowing something out. Then you bring your ribs together. So these are the internal intercostals that are all between the ribs here. And they are used for forced exhalation. They're not the only ones. Abdominal muscles like the transversus abdominis that you can see here are used for that, as well as the obliques, you might remember, and the rectus abdominis. So you can even feel that when you blow out, you're going to use your abdominal muscles to do that. So that's in the interior. Of course, there are muscles on the exterior. I don't have these in order. Let's see this one. Yeah, this one shows them kind of at an angle. So let's share this screen. And there's great pictures for post. I'm trying to click closed all these windows I have open. All right, let me share this one with you. So hopefully you're seeing the torso kind of at a bit of an angle. So here's that rectus abdominis. You can't see the external oblique because it's on the other side, but here's the internal obliques. So these are all forced exhalation muscles when you're blowing out. Here are the internal intercostals that would work with them. So then this kind of pale group here is the external intercostals. These actually contribute 30% of your relaxed inhalation. They certainly would get involved if you're forced inhalation as well, but your diaphragm does 70% and 30% are these guys just when you're uncautiously breathing. If you go to do a forceful inhalation, then we have the pec major, the pec minor. Those are important for lifting your rib cage. And we're just seeing at the very bottom here, I think I have some other pictures of the scalenes. So you can see how they're attached to those first ribs. So they're going to lift up your chest wall. And the not shown here, sternocleidomastoid. I believe I have a half head that shows you that you remember the big muscles that go around the side. So if I stop sharing this, hopefully I won't close you when I close everything else. Let me get into those guys again. That's the chest wall, upper torso, head. Yeah, so here we have that same model. Let me share the screen with his, the scalenes here that are going to these upper ribs. You're seeing them just peeking underneath the clavicle there. And I took away from here, I'm sorry, it's a little off the normal setting, but this is the sternocleidomastoid, but it's missing from that side. I took it off when I took the picture. So when you go to breathe in forcefully and trying to do your vital capacity, you're going to use the sternocleidomastoid together, and you're going to use your scalenes on each side together, and then your external intercostals. your pec minor and your pec majors are all going to lift the rib cage maximally. Okay, are there other models that any of you guys are struggling with that you need any help with? If you had a chance to look at them. Let me look at this one, see if that one's worse. Yeah, this one maybe is worth showing, but I did it verbally. Share screen. This is the older lung model where it's easier for me to point it out. Oops, I'm sharing the wrong one. Sorry. Stop share. Why did that pick the wrong one? This one. Well, maybe it isn't. Maybe it's just hidden under there. Okay. Do you see the lung now? Let's see if anyone's giving me a thumbs up. Hopefully you see the lung because that's where we're going. So I wouldn't ask left and right here, but you could see 1, 2, 3 lobes. Can see the fissures here that are defining the lobes. So this one must be the right lung. What I'm pointing out is the hilum here. So here are the primary bronchi. They're already branched, in fact, into secondary, but that's the bronchi coming in. And then this is the pulmonary circuit. So don't forget, pulmonary circuit, that means that these are veins and these are arteries. So don't go by the color coding. Go by the knowing this is pulmonary circuit. Okay, for the exam, can we shorten? Yes. If someone was asking about the abbreviations, so sternocleidomastoid could be SCM. If we use that abbreviation last semester, you're welcome to use it again. You could say internal intercostals, you could write INT, intercostal. Just if it's your unique abbreviation, then you better tell me what it is, because obviously, I don't know what it is then. So, yeah. So you've got a lot on your plate. I do want to give you some time to work on this, and then you can shout for me. I didn't go through, I guess, all the answers of this lecture questions. So maybe I'll quickly go through those. And then I did answer some of these last time. So if anyone would just go off and work, they can, or you can listen to me drone on. So number 4 asks about structures that fulfill immune functions. Well, I mentioned the tonsils with their crypts, the mucus and saliva, which you should know the components of what's in them, like mucin, defensins, IgA, lysozyme, water. We talked about reflexes like coughing and sneezing, and using pseudostratified with cilia and moving things to the esophagus so you can swallow it. For number 5, 4 unique functions of the larynx beyond the warming, moistening, and filtering of air, preventing food from going into the trachea. And that's done by this epiglottis speech. We talked about the true chords, cartilages that keep the airway open, closing the airway with your vestibular folds. So there's 4 of them there. Describe the structures that enable you to alter pitch and tone. Well, you have voluntary muscles whose name you don't have to know on their retinoid cartilage that changes the length of the true chords, so that longer length you have lower tones and shorter length you have higher pitched tones. Functions of the trachealis, I mentioned last time, is that ring, remember, of the cartilage. Oops, let me stop sharing the screen. That ring of the cartilage. If you narrow that gap between the ring by contracting your trachealis muscle, Airspeed will increase when you do something like cough or sneeze. You might want that to happen. And then if you're just sitting here and you go to swallow food, it actually does a relaxation so that food or food can kind of, a bolus of food can kind of poke into that empty space. Anatomy of bronchi versus bronchioli. Why the bronchioli are more important in regulating? Well, because the rings become plates and are replaced by the smooth muscle. And then number 9, the neurotransmitters we talked about, acetylcholine on muscarinic, which are excitatory. And then histamine also excites that smooth muscle, causing the diameters of bronchioli to get smaller, whereas norepinephrine, epinephrine act on beta-2 receptors to cause relaxation of the multi-unit smooth muscle that makes the muscularis externi. Okay, so are there any questions besides the ones that were asked? I know I zipped through that. I think the only question I could think I skipped was the number 1 of the respiratory physiology, but I answered it. It asks about naming the muscles of relaxed inspiration, and forced expiration. That's the first lecture question of the physiology set, and we talked about them already. All right, so how about for the last half an hour, I turn you loose. If somebody wants to be in a breakout room, let me know. And you guys can work together on these models and try and correlate them to the pictures in the lab manual. And otherwise, I will see you on Thursday. And we are going to be doing spirometry. There is a handout I loaded, I think yesterday or Sunday, since there's the video from Kevin, and then there's a bunch of pictures from Kevin of data. So I'm going to be talking about a lot of the theory of spirometry in the beginning, and the restrictive obstructive diseases, and how you use spirometry to help support a diagnosis of one of those diseases. So when you come back on Thursday, have those things together, because we're going to work on those. So you hopefully will also notice, if you're feeling a little like, oh my God, when am I going to get time to work on all these histology drawings and models, is that you have a lab. We do spirometry, and then we actually have 2 labs before the lab test. And the slides are due that first Tuesday, the 27th. But you have another whole lab where I don't have anything except review or study time for models. So you have kind of another whole lab that I won't have anything much to say on the 27th. For you to work on these slides, but let me know if you're having any difficulty, and I will come and help you. Okay, if anyone needs anything, then just go ahead and shout out. I'm here, I'm not too far off if I get up, so let me know. All right, otherwise I'll see you on Thursday.